everyone. Welcome to EU-OPENSCREEN Training School. Good morning and good evening to those who are watching us and they are in different time zones. Welcome again to EU-OPENSCREEN Training School, which is running for the 4th year in a row. We are happy to have attendees from all around Europe, but we are happy to also having with us people from Brazil, Korea, Africa, US, India. Welcome again. It has become an annual training event which is currently running online. Thank you for being here today. I'm Kathy Skopelitou and I will be your host for the training school. Of course, I would like to say a few things, and I would like to say a warm welcome to all our partner sites, member and observer countries, included the newly added chemoproteomic sites, the Technical University of Munich, the Hochschule Mannheim Center for Mass Spectrometry and Optical Spectrometry, and the Karolinska Institute Chemical Proteomics Core Facility in Sweden, who have just joined EU-OPENSCREEN. As well as the new chemistry site, which is the Medicinal Chemistry Department of University of Barcelona, and the new screening site, the Institute for Research in Biomedicine in Barcelona. Just before we start, I would like to say that our training school will be running for the next 3 days. We will start with 2 morning sessions from 9:30 to approximately 11. Then we will have a coffee break for 15 minutes just to recharge our batteries. And then we will have another talk before the lunch break. Then after an hour, we will continue with our afternoon sessions, which will be very interesting and will run approximately until 3 o'clock. Some organizational guidelines now. The talks will be recorded and each talk will last approximately 40 minutes. Then we will have a live Q&A session of approximately 5 minutes. During this session, you can just raise your hand and ask your question directly to our speaker, or just type your question in the Q&A box at the bottom of your screen. At the end of the talk, I will be able to unmute you and you can ask your question directly to our speakers. With your participation, our school will be more interactive and enjoyable for you. So coming to our agenda for today, we will start our school with a talk of EU-OPENSCREEN Director General Philip Gribbon, who is going to give us insights into EU-OPENSCREEN ERIC and its services. Philip is known to many in our community for his time as EU-OPENSCREEN project coordinator during the later part of the organization's preparatory stage. Phil helped to establish EU-OPENSCREEN as the leading European research infrastructure in chemical biology and supported it to the legal designation of ERIC in 2018. Currently, Phil is head of discovery research at the Fraunhofer Institute for Transnational Medicine and Pharmacology in Hamburg, where he leads several activities in data-driven target validation and early drug discovery. In a 30-year professional career spent in academia, pharma, biotech, and non-for-profit research, Phil has contributed to academic and industrial programs across many areas, from screening infrastructure technology development to drug discovery for rare diseases. In addition to his day-to-day responsibilities, Phil also serves as Vice President of the Society of Lab Automation and Screening. In his talk, our attendees, and especially the young scientists, to whom our training school is mainly dedicated, will have the opportunity to learn more about EU-OPENSCREEN and its great services to biologists, chemists, and the scientific community in whole, as it contributes to the wealth of open, fair screening data publicly available to all scientists. Phil, welcome. We are excited to have you here with us today, giving the opening talk for the autumn's training school. So if you are ready, you may share your screen with us.
Okay, thanks. Sorry, can you hear me okay, Kathy?
Yes.
Okay, super.
Absolutely.
So I'm going to share the screen. Okay, I don't have that option at the moment, but let me know.
Let me see if... Can you see now the green button at the bottom of your screen, which says share?
Yeah, I've got it. That's
Okay,
great.
great.
Thank you very much. Okay, so can you see my screen okay?
Just a moment, it's coming up. Yes, that's great. Thank you,
Okay,
Phil.
thanks very much. Okay, so hello everybody, and thanks for the kind introduction, Kathy. So today I'm going to be talking a little bit about EU OpenScreen, which is the European research infrastructure for chemical biology and screening, and talking a little bit about how we can enable the drug discovery process through access to the research and chemical biology infrastructures that we have. But before I start, I just want to reinforce the fact that what we do here is an open science activity with EU OpenScreen, and it's essentially driven by data. And data is essential in drug discovery, and in modern drug discovery, it's a data-driven process. So since 1972, people have been talking about computational-based drug discovery. Since the early 1990s, the AI is being used within drug discovery. So it's not just within the last 2 or 3 years, and then even more recently, quantum computing-based drug discovery has been used. So drug discovery and screening and chemical biology is a data-driven process. And it's absolutely the case to say that discoveries are made using shared data. And that means that data has got to be discoverable and readable by humans and machines, and it's got to be able to be accessed and retrieved, and so that it can be described so that others can make sense of it. But what we find within my organization and within many of the organizations involved in drug discovery is folks spend an inordinate amount of time working on the preparation of data and the analysis of data, and compared to, let's say, experimental work which is going on. So yeah, so that's it. It's a little bit delayed. So in order to sort of facilitate that process of reusing data, something that's extremely important to the EU OpenScreen community is the application of fair data and fair data principles. And this is where we want to make the data that we're generating within our community available for reuse to others wishing to initiate their own probe or drug discovery program. So that means we have to make the data findable so that it can be easily retrieved and that we can connect data. And we have to make it interoperable. Bioactivity data is just one type of data. There's many other data types which are relevant to really understanding the role of a signaling pathway or the role that a compound may play in terms of modulating a disease process. And for instance, we need to link to imaging data sets, through the structural biology data sets, through the metabolomics data sets, proteomics data sets, etc. In order to have a complete view of the action of a compound. And also data needs to be accessible. So there needs to be a way to contextualize that data and also facilitate its reproducibility. But in terms of access, we need the data to be as open as possible, but as close as necessary. So for instance, clinical trial data is not necessarily open to everybody because of regulations around patient privacy and GDPR, etc. And then also we want data to be reusable. So there's sort of clear license terms on how people can reuse that data. And the reason that we want to do this is because the process of drug discovery is a complex process which involves transfer of information across and between organizations. And there's churning membership, there's people coming and going. So if there's not a readily accessible corpus of data available within each of the key players during that drug discovery process, it's going to make it very, very difficult for that to happen and for that to be successful. So you want to avoid knowledge loss. So that's why we as a research infrastructure are committed to the FAIR principles and making our data available in a fair manner. And just to be sort of completely open with everybody, fair is not the same as open. You can have a fair data set which is only restricted to use by individuals within an organization, let's say within a pharmaceutical company or a biotech company, or you could have a completely open data set as well. And you don't, as I mentioned before, there's issues around individual level person identifiable information, genomic data, etc., the requirements of GDPR, and the rights and roles of research participants and patients as well. So in not all cases can data be open. So just to help with this process of making data fair, something that we contributing towards is the creation of the FAIR Cookbook. And this is an online resource, and I'd encourage everybody to take a look at this if you're interested in working with data. This provides a collection of recipes that cover the practical steps involved in making your data resources fair. And that's something for those who are maybe not data scientists themselves, or maybe just regular research scientists who want some practical guidelines. It's got a number of recipes that help you increase the findability, accessibility, and interoperability of data sets. And this work has come from the combined efforts of many pharmaceutical and biotech companies as part of an innovative medicines initiative project that we were involved with as well. So I would encourage people to to look at those resources in parallel to what you're learning from the EU OpenScreen autumn training, to give you a wider perspective on how to improve the reuse of your data sets. And there's some associated publications that you will be able to find online as well, and also webinars related to that. So there's some additional training material for you there. And what can you do with this FAIR data? Well, actually, then you can get into the world of interlinked, interoperable, semantically interoperable data sets. So here's a data resource where we're linking all of the information associated with various pathogens and all of the compounds which are active against those different gram-positive and gram-negative and fungal pathogens. So these are data from public data resources, and we can link the information about the compounds, the compound structure, publications around the compound, and then we can have a complete view in an easy visualization of the activities of those compounds. And we can also do the same for targeted protein degradation. So those who are interested in this as an alternative modality to the sort of classic inhibition, and we've been able to, using FAIR data resources, we've been able to create knowledge graphs for PROTACs and molecular glues and degraders. And in this case here, we can show the for the androgen receptor, we can show all of the compounds, that we can show the diseases which are associated with the androgen receptor, the signaling pathways, which PROTACs bind to the androgen receptor, which E3 ligases are part of those PROTACs, which warheads are binding to the androgen receptor. And we can link all of that information together and come up with useful insights and new information about the different roles of PROTACs and molecular glues in disease. And then we can use that basis as the basis for machine learning and artificial intelligence algorithms to make predictions about PROTAC and molecular glue properties. So that's just some sort of side information on working with data and FAIR data working and what can come out of it. So I'm going to skip now to EU OpenScreen and what we're doing there within that research infrastructure. So we as a research infrastructure, we provide access to a range of technologies, for instance, screening platforms, high-throughput screening platforms, resources, and it's an extensive range of chemical libraries which are available for access by users at our partner sites. Specific expertise in, for instance, in medicinal chemistry. So oftentimes many probe and drug discovery projects start and finish with finding a hit from a screen. And it's not so often that those hits are subsequently optimized towards leads and advanced leads, preclinical candidates, and candidate compounds. So we have that expertise in medicinal chemistry to enable that process. And as I mentioned before, we're also very much in the business of generating large open data sets, FAIR data sets which can be reused, of bioactivity data related to specific on-target, off-target, and phenotypic results. And as part of what you're involved with today is we're training individuals both within our community, but also within the wider community to be able to access and use these resources and be able to progress their own projects. And we do that in collaboration with academia and with industry as well. Kathy's already shown you the our map of country sites at the moment, and I think we've for those who maybe joined us last year, we've actually got 4 new partner sites this time, actually five new partner sites. And one of the most interesting categories is joining our regular screening partner sites where high throughput screening is performed. We also have 11 chemistry partner sites where optimization of compounds take place. But now, just in the last few months, we've been joined by 3 new chemoproteomics and spatial mass spectrometry-based omics partner sites. And in those partner sites, we provide access to technologies that allow scientists to be able to better understand the mechanism of action, for instance, of compounds which have come up in a phenotypic screen. You can use chemoproteomics techniques in order to find which targets were involved in the phenotypes which were being observed. And with spatial mass spectrometry, you're able to look at the distribution of your compound in different tissues and different cell types, and that allows you to try to better understand its pharmacokinetic and pharmacodynamic properties as well. So we're a growing community which is extending its range of services which is on offer. Okay, so yeah, so the EU OpenScreen is part of a wider landscape of research infrastructures, and we collaborate with them intensively. So those are research infrastructures which are focused more on, let's say, structural biology, so looking at the structures of proteins, or imaging technologies, especially, and that's the Eurobioimaging community. There's also the structural biology with the Instruct community, or clinical trials, which is the ECRAN community. So we sort of operate within a continuum of life science research infrastructures that are able to offer services and access to technologies and expertise and know-how, going all the way from the basic understanding of the role of a particular target or pathway in disease, all the way through to the point where you can take compounds into the clinic. So the access model involves we see 3 main types of organizations accessing our services. So they're either biologists, so those are individuals who are bringing, or individual scientists who are bringing targets onto the platform, and they want to run screens and identify hits from that. Chemists who might want to donate their compounds, so that they can also be screened at the different partner sites, or data scientists. And the categories of partner site are screening, chemistry, and database. And also we just recently added to that the category of chemoproteomic sites. And EU OpenScreen is, as a centralized office, we provide the compounds. So we have the compound library and the logistics around that, and also training, and also helping work with the database side of things. So in terms of our libraries, we have the largest one is the European Chemical Biology Library. This is nearly 100,000 compounds which are structurally diverse. So these are typically drug and lead-like compounds. In addition to that, we have a pilot library of 2,500 compounds, which represents bioactives typically against around 1,000 different targets, and these are highly annotated. We also have a fragment library of low molecular weight compounds, and around about 1,000 of those are good for biophysical-based screens. And then also we have around 6,000 compounds which have been donated by chemists from across Europe. We perform quality control on those, and then those compounds are put into the screening collections. And the idea is that we're able to, through screening those compounds at partner sites and user projects, we're able to build collaborations between chemists and biologists around targets that the compounds may not have seen previously. One important activity within the EU OpenScreen are bioprofiling assays. So this is where we've taken our 100,000 compounds and also the compounds submitted by academics, and we subject them to a range of assays where we assess things like the solubility of the compound, because a compound which is insoluble is unlikely to have a significant biological action. So we understand what that is, but we also look at assays which are able to predict common interference systems within screens. So for instance, compounds which lead to the generation of reactive oxygen species, compounds which interfere with bioluminescence and other readout technologies. And an important point of that is also cell painting, which is a phenotypic assessment where we're trying to generate phenotype to phenotype relationships. An important part of what we do is also the centralization of the data which is generated within the projects. So at the moment we have data in the European Chemical Biology Database, and then we recently published a paper on nucleic acid research which describes that in detail. So I would encourage everybody to look for that paper. but we've got about 5.1 million data points at the moment, and 3.6 million of those data points are actually available for download and for reanalysis by members of the community. One example of such an analysis of the data was a recent competition that was launched in Kaggle. And in this competition, what we did was we looked at our solubility data set. This is where for the 100,000 compounds in the European Chemical Biology Library, we assessed the solubility at one of our partner sites. We were then we provided 70% of that data to the community, and then asked them to see whether they could, using that data and that information, whether they could predict the solubility of the remaining 30% of the compounds which we'd kept as the test set. And we were actually able to run that quite successfully. There were over 100 teams were interested in analyzing that data, and we published a series of articles earlier this year where we described that challenge. We described the algorithms that were the most successful in being able to predict the solubility of the compound. And this is just one example where the providing fair data sets enabled the improvement of the overall understanding of what the molecular determinants are, the chemical determinants are of compound solubility, and that should help also in the future design of libraries for screening. So finally, I just want to briefly touch on another extremely important activity that's taking place, and this is our cell painting initiative, and this is being led by my colleague Christopher Schmid, and also colleagues from the FMP, the Leibniz Institute for Molecular Pharmacology, based in Berlin, which is where we're also co-located as well. And within this this exercise, what we're looking to do is to perform cell painting. So in a classic target-based drug discovery, you have a target-based assay, you identify compounds which bind to it, you generate leads, and then you optimize those leads towards clinical trials. In phenotypic assays, you typically have a disease model represented in your in vitro system. You find a compound which modifies that disease model and produces an efficacious result. You then select that lead, then you go through a target identification process using typically chemoproteomics, then you go into lead optimization. So within the cell painting paradigm, what we do is we look at perturbations to different morphological signatures within the cell. So we stain the endoplasmic reticulum, the nucleus, mitochondria, etc., with various stains, and we look at changes in the cellular morphology upon treatment with compounds, and we try to use those changes to try and map through to what the mechanism of action of the compounds might be. So we produce fingerprints for compounds of known mechanism of action, and then when we have new compounds of unknown mechanism of action, we compare those fingerprints, and that allows us to come up with predictions of what the phenotypic hit mechanism of action may be. And we're undertaking one of the largest cell painting efforts globally, so we're going to be working on over 107,000 compounds overall. That work is pretty complicated. We've got to do typically we're only able to generate sort of 5 to 10 plates worth of data per site each day, and it involves multi-dimensional imaging. We're generating 9 different image fields per well, and in 5 different channels, where we're looking at the nucleus, endoplasmic reticulum, and we're looking at the cytoskeleton, the mitochondria, and the Golgi as well, all in parallel. And this work is taking place at 4 different partner sites, and the challenges we have are around standardization of those data and making sure that we can compare the different data sets on the different partner sites. Each one is screening 25,000 compounds. And this is the type of images that you're getting. In our case, we chose HepG2 cells, but also we're working on U2S cells. And in some initial testing on our bioactivity data set, we generated nearly 2 million images already. What we're doing within the data analysis process is that we're reducing the number of image features. There's an enormous number of different image features you can extract using programs like Cell Profiler. And we're reducing that down to 570 or so, and then that generates these profiles that you can see down on the right-hand side. And now we're right in the middle of this whole cell painting effort. And between now and the end of 2025, we're about halfway through that. We're going to be generating cell painting data sets for each one of our 100,000 compounds, and we intend to make those available, both the primary images themselves, plus the analyzed images, plus the workflows, and they'll be available for reuse by anybody within the community, and they can be used in parallel to other data sets which are being produced by organizations such as the Broad Institute, etc. We're going to invite everybody to Berlin. At the end of November, we're going to have a hackathon to analyze those cell painting data sets for those 100,000 compounds, and also in combination with other data sets as well from other partners. And finally, I just want to invite everybody who's interested to join us at the International Chemical Biology Symposium and our European Chemical Biology Symposium joint meeting in Paris, which is going to be happening in October. And we've got a stellar range of speakers from both Europe and the US and elsewhere. And we'll be looking forward to seeing you there and talking about chemical biology and screening and building connections with between you and the rest of our team at EU-OPENSCREEN. So thank you very much. And I think I'm finished.
Many thanks, Phil, for this very useful presentation. I think we learned a lot about the EU-OPENSCREEN, but also cell painting assay and FAIR data. So let's go to the questions from our attendees. I can see that one of our attendees, Hamed, has raised his hand. Hamed, can you hear us? Okay, let's try again later. So yeah, maybe I have a question, Phil. I know that a lot of chemists and biologists right now are watching us. So what would be the first steps to get in touch with EU-OPENSCREEN and start their project?
Yeah, so I think it's basically we have forms on our website, and just reach out to us. And there's a very, very simple form to fill in where you just basically say what it is you're seeking to do, run a screen, donate some compounds, get access to some data. And then if you just fill out that form, we'll connect you directly. So if you go to the EU-OPENSCREEN.eu website, you'll see that there. And we're very happy to talk to people. And I think one of the things we really realize that it's expensive to perform the kind of work that we're involved with. And we're also happy to assist organizations and individuals and provide them information for their grant proposals so that they can it's a competition of ideas, and we know that, and we want to help you get the resources and funding that you need in order to come and work with us.
That's great. I think you can see the question from John in the Q&A box at the bottom of your screen, but I will read it in any case. Yeah. Is the hackathon mentioned on the second last slide being an in-person event only?
It is an in-person event. So we're anticipating around 100 people will be able to come to Berlin, and we'd encourage you to. We've just come out with the initial announcement today. We'd encourage people to come along, and we're setting a data infrastructure at the site, so we will have access to all the data plus other data sets, attendant data sets, and we've got some really exciting data. So, for instance, there's a genetic screen that was run at the Broad Institute and in a similar setting in cell painting. So we'd like to be able to connect results from genetic screening to results from target-based screening and see how that looks. So there's all sorts of exciting and interesting questions that we can ask of those data. So we'd really, those who are interested in cell painting, image analysis, establishing chemotype-phenotype relationships, please come and talk to us and meet us in Berlin next year.
This will be definitely a great event. So let's go to the next question from Tim. With the cell painting project, are there plans to use other tissue-specific cell lines to test compounds?
So I think in the case of some of the active compounds that I think typically at the moment, without wishing to sort of pre-announce what the results are, but typically only about 5 to 8% of the compounds which we screen are actually creating some kind of modifying phenotype in the cell painting assay. So there's an opportunity to take some of those compounds and screen them in other cell lines to see whether that's maintained, because we absolutely understand that doing too much of a detailed analysis in one cell line doesn't necessarily give you a generalized picture. We need to look at how these compounds perform in different cell lines. So there's a lot of expertise in different cell lines. We're doing one cell line. There's other members of the consortia have access to other cell lines. So we will be following up the compounds with interesting activities. Absolutely.
Great, thanks, Phil. So just the last question before we finish with your presentation. So from my side, I know that you have presented the ECBD database in one of your slides. So could you please clarify to everyone, is it free? Is this database free? Can someone download data from there?
Yep. So, I mean, it's free in the sense that public organizations and countries in the European Union have paid a lot of money for us to create this resource and make it available and make it fair, especially, and also to generate the data that's in there. But for the users from the scientific side, it's absolutely free to use. So anybody's free to download all of the data that's in there. There is some data which is from our programs, which is under an embargo. So we have an embargo of up to 3 years, and that allows organizations which have run screens to then, further optimize compounds, generate IP, etc. And you saw there's about 1.5 million data points under that embargo. But in all the rest of the data is free to access, and that embargo data will eventually become free to access as well. So yeah, it's all free, and we're currently working on actually improving the process of accessing the data, so the data are better linked to machine learning based workflows, so that folk can port the data directly into their machine learning workflows as test data sets to build models around. And we want to facilitate that model building process.
That's great. This is a great opportunity for everyone to have access to all this data. So at this point, Phil, I would like to thank you very much for this interesting talk. And yeah, I think we will have the opportunity in the near future to present more about the EU-OPENSCREEN and its services. Thank you very much.
Thank you. Thanks, Kathy.